Okay. Part 3, Chapter 22. Fifth factor for the speed of reaction is the presence of a catalyst. And what a catalyst is a chemical that is not a reactant or a product. It's not consumed in the reaction, but it does take part uh, in the activated complex. It helps the reactants to come together at the right time, right place, correct angle on the functional group with enough energy to break the bonds. And what it really does mostly is assist with orientation. But once it's done doing its job, it's free to release the reactants, the activated complex rather, and go along its merry way and help other chemicals. So let's see what we got as far as our notes. All right, textbook definition to mesmerize. Catalyst, a chemical substance that lowers the activation energy and speeds up the reaction, and that's the key here. We're looking at lower activation energy, which means alternative pathway. Alternative pathway. In other words, we're going to come up with a different way. Looks like we're going to come up with a different way of spelling alternative. Looks like an F. Alternative. We're going to come up with a different way of the reaction proceeding, and that's going to give us a different energy profile. All right, so there's two different types of catalysts, homogeneous and heterogeneous. The homogeneous becomes part of the activated complex. The heterogeneous really doesn't. It provides a surface for the reaction to occur. And when you talk about catalysts, you should also talk about chemical inhibitors, so we're going to throw that in as well. Heterogeneous catalysts uses adsorption. Now, adsorption. Ab means to become part of the structure. So a paper towel absorbs water, gets into the structure. Add means it sticks to the surface. So you step on gum, for instance, in your shoe, it would stick to the surface of the shoe. It wouldn't become part of the sole of the shoe. So adsorption, we're going to stick to a surface. That's going to provide a location for the reactants to come together, and that is going to allow the chemicals to orient themselves so that they can form products. Right. So catalytic converter, that's a small part of a car in the exhaust system, and it was added by environmental pressures. Pollution was causing something called the acid rain in the 80s, and what would happen is you would get things like carbon monoxide and nitrogen monoxide and nitrogen dioxide coming out of the tailpipe, and that would react with the H2O in the water. In the next unit, we're going to talk about this reaction, this hydrolysis reaction, that produced an acid, and that acid would come down in the form of rain. Now, a catalyst is something you attach to the exhaust system. It has platinum metal on the inside. And what the platinum metal does is it provides a location for the chemicals to come together and react. Now, we're talking about gases. So without a catalyst, the gas would pass through the exhaust system. But if some of the gas that's produced from the exhaust waste sticks to the platinum metal, there's a possibility of a reaction occurring. So suppose we had something like this. Suppose this black checkerboard was the surface of the platinum. So I'm going to label it PT for platinum. And let's say our gas is moving through a pipe from left to right. Say we have a pollutant like nitrogen monoxide, NO. Now, normally these molecules would just hit each other and bounce off. But if nitrogen monoxide is passing through the tailpipe, it could stick to the surface of the platinum. So that would put a little nitrogen monoxide particle here. And then maybe a microsecond later, another nitrogen monoxide particle, which is passing through, gets attracted to the surface of the platinum, and then it sticks right here. Well, now these two molecules that would normally just bounce off of each other or would just pass by each other, now they can react. So maybe these two nitrogen atoms react and become nitrogen gas, which makes up 78% of our atmosphere, and the two oxygens react and become molecular oxygen, breathable oxygen, and passes through the tailpipe. Now, do understand, not all of the particles stick to the platinum metal, but it reduces the amount of pollution, and it has had a big impact in some areas. You still have photochemical smog and things being produced in areas of high traffic volume, but it has helped. All right, so surface catalysts. The catalyst is a solid in this case. The chemical reactants are gases, so we're looking at heterogeneous reactions. So the catalyst provides a location, a surface for the reaction to occur. Now, homogeneous catalyst is something in the same phase as the reactants and actually bonds to the reactants for a brief moment in time. Say that it assists in bringing together the reactants. So it becomes part of the activated complex. And what it does is it helps with the orientation of the reactants. The reactants, let's say you have a protein or a large macromolecule, thousands of atoms, and you only have certain functional groups. Well, chances are most of your collisions are not going to be on the functional group. I mean, particles are moving randomly, so there's only a random chance that you're going to hit the functional group. But with a catalyst can help aid the orientation of that and make sure that the functional groups come together. The key to understanding homogeneous catalysts is that it lowers the activation energy. It's providing a different way for the reaction to occur. Instead of relying on random particle collisions, the catalyst is going to attract the reactants and bring them towards each other in a way so that they're oriented properly. Pardon me. All right, so these two videos. The first video, I really just want you to watch. If you go to the link, I don't know why I'm getting this echo. For those of you who are watching it in the spring, hopefully by the summer I will figure out what the problem is and you won't have the echo. But I really only want you to watch the first 2 minutes and 6 seconds of the first Catalyst video, and then 2 minutes and 15 seconds. I think both videos are like 6 or 7 minutes long. I'm going to show them now, but again, I'll provide the link so you can hear them without the echo.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed.
All right, so I'm going to stop it there for a second. Again, that's the key. Two keys right there. One, that we're not talking about speeding up a reaction a little bit with an enzyme. We're talking about speeding it up a tremendous amount. Type of reactions that would be so slow based upon random particle collisions could not occur fast enough in the human body to keep you alive. So enzymes are biologically very important. That whole thing about temperature and pH. Again, we could raise the temperature to get something to react faster, but we would have to raise it tremendously to have the impact that a catalyst has. We couldn't survive those temperatures. Same thing with pH. Our blood is slightly basic, like that little OH minus in there. But at high acid concentration, again, we wouldn't be able to survive, even though that might create an environment that speeds up the reaction.
To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products.
I just want to stop the video because look how happy the products are. Now, all that smiley face. Before, when they were uncatalyzed, they were having trouble getting over that activation barrier.
In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products.
Unhappy reactants. Happy when they're products.
An enzyme acts as a template
the enzyme.
Binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier
Look how happy.
for the reaction to occur.
Oh, absolutely hysterical.
To understand how enzymes work,
so that
let's
my
take a
humor will be
closer
funny.
look at a reaction in the citric acid cycle.
No, let's not. All right, so 2 minutes and 6 seconds. Good for us. All right, so again, the keys there. That energy diagram at the end, we're providing a different pathway, a lower energy activation in order to increase the success of the collision. Second, we can run this at low temperature. We can run this at mild pH. Alternative pathway.
Enzymes are proteins which catalyze metabolic reactions involving the conversion of a substrate into a product. Enzymes bind with substrates due to their complementary shape. This is called the lock and key model.
Okay, I got to stop here. Actually, in the past 5 to 10 years, they've proven that this lock and key model thing doesn't actually happen. The induced fit one that they're going to talk about, induced fit, that is actually what happens, that the enzyme actually changes shape a little bit to accommodate and work with the substrate. So again, this lock and key, they don't even like to use the term anymore. It's very misleading. There's no lock and key here, but there is, if they want to say complementary shape, meaning that the shape helps with the function, then I think that it's okay to say that.
Rather like a spaceship docking into a space station. In the induced fit model, the enzyme changes shape to accommodate the substrate.
And this is the accepted model now. Now, substrate just means reactant.
Enzymes enable metabolic processes to proceed rapidly at room temperature by providing an alternative reaction pathway of lower activation energy. Without enzymes, these reactions would happen too slowly to sustain life, or would require extreme conditions of pH or temperature. Carbonic anhydrase catalyzes the following reaction between carbon dioxide and water. This reaction proceeds 10 million times faster with the enzyme catalyst than without. A water molecule is coordinated to a zinc atom in the protein. A proton is removed.
Okay, so it's only 1:33. I had that one wrong. It was 2:16 or 2:17 for the whole video. Oh well, oopsie poopsie. Okay. So again, the substrate, those are my reactants. And the enzyme is going to be my helper. The reactants are attracted to the enzyme, and that changes the orientation and the shape of the reactants, and that allows them to produce products. In this case, we're looking at a decomposition. But here's the key. It would take a look at this energy profile. So on a multiple choice test, they would ask you, well, without being labeled, which of these represents the catalyzed and uncatalyzed reactions? And of course, the one with the lower activation energy is the one that we would say that, all right, here's my catalyzed reaction. And again, alternative pathway. The energy of the reactants is the same. That's important. The energy of the products are the same. So therefore, the delta E, change in energy, or delta H, the enthalpy, is still the same. It doesn't change the enthalpy of the reaction. What it does change, however, is the activation energy in the activated complex. All right, so two types of catalysts. Again, homogeneous catalysts form part of the activated complex. They can be run at mild pH and for enzymes at regular room temperature or body temperature, and that they speed up reactions tremendously. And what else did we say that was important? Provide an alternative pathway with a lower activation energy. Heterogeneous catalysts, again, we're providing a location for a reaction to occur. Okay. All right, so again, reactants known as a substrate bind to the enzyme. That changes the substrate, it also changes the enzyme, and what we mean by that, induced fit, and that allows the reactants, in this case, to decompose and break apart and produce products. And we said make sure that we understand the finer points here of the energy diagram, that delta H is the same regardless of whether a reaction is catalyzed or uncatalyzed, because delta H is the heat of the products minus the heat of the reactants, and that does not change just because we have a catalyst. What changes is, again, our activated complex. Okay, so the second topic I want to talk about in this video is reaction inhibition or inhibitor chemicals. First of all, a definition for us to mesmerize for a test. Inhibitors do not slow down reactions, they stop them. In other words, another name for this, like I said in the other video in organic chemistry, we call this a quench. We can add this. Usually it's something that changes the environment, and the environment is no longer conducive for the reaction to occur. That's one way of inhibiting. Another way is to actually become part of the reactant. And we'll talk about that in just a moment. A very short moment. For instance, in cell signaling in biology, you have a signal molecule that is produced. And let's say it travels across a synapsis and into a receptor site. Well, this triggers a chemical response by the cell, and that chemical response can get into the nucleus here. The molecules get into the nucleus, and maybe this tells part of the DNA to produce a certain protein. And then let's say that this protein could be something helpful, could be because many helpful biological processes are controlled by cell signaling, but it could also be something harmful. Let's say, for instance, the signaling molecule is a virus, and that virus can tie up that receptor site and then trigger a chain reaction of chemical responses that produces a protein, and maybe this protein is what we need for instance, for bacteria to grow or something of that nature. One of the ways that we can combat this is if you get a chemical of similar shape and it ties up this receptor site, it can block this chemical process, and so that it doesn't signal the DNA to produce this protein, or the nucleic acid to produce this protein, and then stop that protein from being produced and signaling other cells So, we call this competitive inhibition. We can get another chemical to tie up a receptor site or to tie up a molecule so that it blocks a series of chemical reactions. So really, and there are other types of inhibition as well. There's non-competitive inhibition where the inhibitor chemical actually bonds to a different part of the molecule rather than the receptor site, and it doesn't matter whether the signal molecule gets there or not. But we're not going to get into detail on this sort of thing. Again, just to say that inhibitors can be really important biologically. If we could tie up a receptor site, we could stop the series of chemical reactions from occurring in the cell. And I used two examples of how competitive inhibition can be beneficial here. One is in tuberculosis, that showing it in green, this growth. It can be blocked again by the filling up the receptor sites. You fill up the receptor sites, the protein that tells the bacteria to grow no longer is produced, and the TB winds up it winds up those bacteria cells wind up dying off. And then the other example, and this was a thing about 10 years ago, this MRSA bacteria. It's funny, the thing that blocked that, one of the natural things, it's just eating your green vegetables, broccoli and Brussels sprouts. And I always thought that was kind of funny because, my grandma would always say, make sure that you eat your broccoli, make sure you eat your Brussels sprouts. And I thought it was terrible at the time. But, hey, grandma was wise. She was, I guess, trying to prevent me from getting MRSA. Anyway, so inhibitor chemicals. If we look at this energetically, if we're looking at a quench, for instance, again, this is an example of inhibition where an inhibitor chemical ties up a reaction site. But if we're looking at this from an energetic point of view, let's make our little graph here. First quadrant, we have energy on the y-axis, and we have time here on the x-axis, and we have our reaction. Let's say we're reacting A plus B, and it's going to produce AB. We might get a pathway like this. Here's my reactants, A plus B. Let's call that reactants. And my pathway might be something like this to get to my products. Now, an inhibitor reaction. Okay, so we're going to throw in a quench. So we'll say, all right, chemical A is going to react with the inhibitor. Let's call it IN. And that gives me this product and stops the reaction between A and B because it ties up and takes the place of B. So it's tying up A. A can no longer react with B. A couple things here. It doesn't have to have both of these, but these are, things that we would look at. In terms of activation energy for the pathway, we may have the same level of reactants, may not be, but we should have a lower energy pathway. And the second factor is we should have lower energy products as well for our inhibitor product, because again, nature tends towards stability. It's more energetically favorable for this product. I'm just going to throw delta G, even though we haven't talked about it here. More energetically favorable to produce that product rather than the one that was originally reacting. Why am I doing that? It is energetically favorable. That's not the word I wanted to use. Thermodynamically favorable. Favorable. Okay. All right, so that's enzymes and catalysts and inhibitors. Next video, I'm going to do the actual mathematics for the kinetics part. We'll take a look at differential rate. We'll take a look at reaction mechanisms. Again, there's a lot in this chapter. So thank you.